Okay, this is going to be a presentation on genetics of cancer. And this is the first of many presentations that we're going to do on cancer. So we're just going to start here. And the objectives of this particular presentation include recognizing the lifetime risk of cancer in the U.S., differentiating between neoplasm, malignant, and benign, summarizing the stages of the cell cycle, and I have to be honest, the cell cycle is the most important thing that you need to understand when you're thinking about cancer. Identify the stage in the cell cycle where mistakes in replication are corrected, discuss the implications of genetic mutations in genes associated with the cell cycle and their implication for cancer, both inherited and sporadic. Recognize the type of cancers and age of onset in patients with mutations of p53 gene, and this is germline, known as Li-Fraumeni syndrome. Recognize the age of onset of gene and chromosomal additional complications seen in patients with retinoblastoma, and identify inherited disorders associated with the cell cycle abnormalities. So cancer is prevalent. About 1 in 3 individuals will develop cancer at some time in their life. And they update this kind of every year, but the main points don't change. The most common cancer in males is prostate, but the most common cause of death is lung and bronchus. In females, the most common cause of cancer is breast, but the most common cause of death is lung and bronchus. And then the third in all these is colon. So, definitions. A neoplasia is a disease process characterized by uncontrolled cellular proliferation that results in a mass or tumor. Uncontrolled or unregulated cell growth results in a mass. Not all neoplasias are cancerous. Malignant, uncontrolled growth is capable of invading surrounding tissues. Travels to other sites through lymph nodes. Metastasize is when it spreads to distant sites, for instance, breast cancer metastasizes to the liver, and benign is a tumor that does not invade or metastasize, but it still can cause health problems. And I'm sure you'll get these same definitions from pathology. Again, types of malignancies: sarcoma, meningeal tissue, bone, muscle, connective tissue, carcinoma, epithelial cells, hematopoietic and lymphoid, what we're going to talk about in hematology, is a cancer in the blood or bone marrow lymphatic system, lymphoma and leukemia. So, cell cycle. The cell cycle is the most important thing for you to understand for cancer. So the cell cycle is always going, and so we have mitosis, cytokinesis, and right after that is G1. And G1 is when all the work of the cell does. Things are being made. Things are being transported. The cell's busy working. Fast-growing cells have a small G1. Slow-growing cells have a really large G1. After the G1 stage, it then goes to S. When we replicate our 46 chromosomes, 3 billion base pairs. And even if we're 99.99% accurate in doing this, there's still lots of mistakes. So G2 is the double check. We make sure we haven't made any mistakes before we undergo mitosis. If G1 recognizes there's a little bit too much damage in this DNA, it moves it to G2, where it just does its work, but it never divides again. The problem with cancer is it likes to skip checkpoints. So there's checkpoints that prevent the cell from going from one stage to the other. And so here's a G1 checkpoint. Is there enough nutrition, growth factors? Is there damage to DNA? If there is, you ain't going on to DNA synthesis. After we, again, replicate our 3 billion base pairs and we double check to make sure there's another checkpoint. Is it the right size? Did we make all our, fixes? And then finally, there's a checkpoint in mitosis to make sure before cell division. So oncogenesis is kind of that balance between growth, proliferation, mitosis, and attrition, cell death, and apoptosis. Lots of factors. UV radiation, viruses, medications, radiation, tobacco, normal cell process, chemicals, and then heredity. These are probably, if not more than this. This just causes it. But all these things are the balance here. This is another really, really important concept, which is all cancer is genetic, but not all cancer is hereditary. Nowadays, we're actually looking at somatic. So somatic is in the cell, inherited is in the Eiger sperm that makes the individual. All cancers develop due to mutations that occur in our genes. But not all of these are inherited. So somatic is something that we acquire, germline's what our genes that we had when we were born. Most cancer is not hereditary, but it's all genetic. Every single cancer has gene changes, but not every single cancer has hereditary issues. So let's just look at this. So here we have the normal cell, and let's say there's 4 factors, and all 4 factors are working just fine. And then you lose 1. But you still got the other 3 to pass you up. Then you lose 2, but you still got the other 2. Then you lose 3, but you still got 1 that's controlling things, and then you lose the last one, and now you're in trouble. That one cell, even though there's millions of other cells, grows so fast it can take over the entire group. Tumor suppressor genes, the function of these genes is to constrain growth, to control it. You don't go too fast, you don't go too slow, you go just the same rate. The loss of function of both copies results in cancer. So both alleles have to be abnormal before you get cancer. So in sporadic cancer, you're born with 2 normal cells, 2 normal genes, and here the divides, most of the cells are normal, but what if we have a change in one of them? We're still okay because we still have our second one. But when we lose the second control, that's when there's uncontrolled growth. So the Knudsen two-het hypothesis is it takes multiple hits before abnormal growth occurs. And this is the concept. We're going to talk about inherited cancers, and they're mostly inherited as a dominant. But you're only born with 1 copy of that gene that doesn't work. So it's like, well, why is that dominant? Since both copies, like in a recessive, like both copies have to be non-functional. And it has to do with the Knudsen two-het hypothesis. So you're born with 2 copies of the gene that works. And then over time, one of those copies, you lose function. But you're still okay because you have the second one. Then you lose the second one, and now you have tumor development. But what if in hereditary cancer, you're born with 1 copy of this gene that doesn't work in any cell in your body? Any breast cell, any ovarian cell, any brain cell, any liver cell, pancreas, so on and so forth. It only takes 1 event before you're in trouble. And then the tumor starts to grow. So inherited cancers, for the most part, are inherited in autosomal dominant fashion. There's a few exceptions to that rule. But if you're going to guess dominant. And the reason it's dominant is you only need 1 more hit. Having said that, the vast majority of cancer is sporadic. 10 to 20% are familial. That means we see a family pattern, but we're not really sure why. And 5 to 10% are hereditary. So sporadic results from buildup of damage to cells over time. Typically, the individuals are older when they're diagnosed, older than 50. They're more common cancers, such as breast cancer in women. And there's no significant number of family members over the general population that are affected. Familial is a combination of genetic environmental factors, maybe lifestyle, not a single inherited gene mutation. And it kind of looks sporadic, but we see more than we would expect in that family size. And then we get to hereditary. This results from an inherited gene mutation that predisposes the individual to develop cancer. Some hereditary cancer syndromes can result from a de novo mutation in the individual in the egg or sperm that made them. So sometimes we don't even see a family history of this, but one of their genes, germline genes, doesn't work. Hereditary cancer syndromes have reduced penetrance. Not everybody loses that second copy. Mutations don't skip generations, but cancer may. Some people get really lucky, or dad died in a car crash when he was 30 and he passed on this gene. He didn't live long enough to get the cancer. There are over 100 different genes we currently know of at the time that I'm giving this presentation, and the number is continuously growing. So this is, don't memorize 100. So I'm just going to talk about a couple of inherited cancer disorders that we understand really well. Fortunately, they're incredibly rare, and one is retinoblastoma. And it's a malignant tumor of the retina, and typically requires removal of the affected eye. Sometimes it's very small and you don't have to, but these are pictures. Here is a kid, you're taking the picture and you see this unusual thing. And here it's bilateral. It's caused by the RB1 gene, which is located on the long arm of chromosome 13. It's a tumor suppressor gene that's important in G1. It's inherited in autosomal dominant and very penetrant. 40% of kids with retinoblastoma are hereditary. When the individual has a germline mutation, it's usually, or a mutation or deletion, there's a significant risk that this retinoblastoma will be bilateral, even if we don't see it at the beginning. Li-Fraumeni is probably one of the scariest inherited tumors syndromes, and it's because of mutation of the TP53. And this is probably one of the most important cell cycle controllers. So mutations or deletions of this gene are often seen in somatic tumor testing. So again, if I do somatic testing, a lot of times they'll have a deletion of this gene, or it's located on chromosome 17, or there's a mutation of it. But that doesn't mean it was inherited. Li-Fraumeni is when you inherit one of these copies you're inherited with. So every single cell in their body has a single mutation. And this particular gene is important in both G1 and G2 checkpoint. So here we have a pedigree of Li-Fraumeni syndrome. And these are the kind of pedigrees it's like, well, in general, brain tumors aren't inherited and leukemias aren't inherited. But look at this family history. She had breast cancer at 35. Sarcoma at 7, leukemia at 6. Her dad is alive, which is shocking, but he has a sister with breast cancer and osteosarcoma, another brother that died of leukemia, mom died of bilateral breast cancer at 40. So, and he carries it, and he just got really super lucky, but, we see very, 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 a huge amount of cancer in the family, and at all different ages. So G2, this G2 checkpoint, Li-Fraumeni, we're going to talk about the breast cancer gene in the next case, and G1, RB, retinoblastoma, and the TP53. DNA repair. DNA makes mistakes, and it has to repair it. Inherited disorders that have to do with DNA repair include xeroderma pigmentosa and ataxia telangiectasia. And unlike most cancer syndromes are dominant, these are actually recessive. And both copies of the POLH gene are not functioning correctly. This is incredibly rare. Clinical features include very severe sensitivity to UV radiation, a high frequency of skin cancers, squamous cell, basal cell, and melanomas. Ataxia telangiectasia is also a super rare recessive disorder affecting the cell cycle, and similar to the last one, inability to repair UV radiation. It's the ATM gene, and signs include telangiectasias in the eyes, cerebral ataxia, and immunodeficiency. So hereditary cancer, clues in a personal and family history. Young age of diagnosis, typically under 50. Multiple blood relatives on the same side of the family. So breast cancer, we're going to talk about, is super common. So if mom's mom had breast cancer at 62 and dad's mom had breast cancer at 70, that's different sides of the family. It doesn't really count for two relatives. Multiple lung cancers in smokers, not as concerning as two young people that had cancer. Individuals with multiple primary cancers, so bilateral breast cancer or breast and ovarian, related cancers in the family, and rare types of cancer, for instance, male breast cancer. So we're going to talk about that more in the next lecture. So if you look at chromosomes or genes of malignant cells, they're going to be abnormal. But in most cases, those are somatic changes. And most cases of cancer are sporadic, and they usually affect DNA and the cell cycle regulation. Most inherited cancers are dominant, except for those last two I talked about that are super, super rare. The second copy of the gene is lost in some cells. That's why only, for instance, 90% of patients that carry one of these genes develop cancer. And there is a chance that they get super lucky. And as I said, xeroderma pigmentosa and ataxia telangiectasia are recessive and very, very rare as far as causes of inherited cancer. And that